Hello, my name is Markus Metzler. I'm pediatric oncologist and hematologist at the University Hospital in Erlangen, Germany. And I have the pleasure to contribute a talk about predisposition and genetic factors in pediatric CML. And to give a short overview on the motivation for this talk is first, of course, to describe the genetic features of pediatric CML, because we do know that there are some differences and we want to explore whether this has relevance for the diagnosis and for the treatment of pediatric CML. And the sources for this talk are mainly a kind of review of the literature and a comprehensive analysis of the German CML pediatric cohort, which I'm chairing with my colleagues. So to give a systematic overview on the topics I cover with this talk, or what I not cover with this talk, is to say that we are generally distinguishing between germline variants, the genetic information which is present in all cells of the body, including the germ cells in the ovaries and the testes, and therefore they are inherited to the offspring. And somatic variants, so genetic information in certain cells of the body, for example, the hematopoietic cells, which are transmitted to the offspring of these cells, so like blood cells, but not to the next generation, to children. And obviously, both these variants, they may have impact on the disease itself, or they may have impact on the course of the therapy. The whole topic of somatic variants influencing therapy, for example, the mutations of the ABL1 gene, this is a very topic by its own, and this has been covered by other talks, and therefore I will not mention this. And also the topic of somatic variants with prognostic significance. This is an upcoming issue which is just to begin to be understood, but in children we have not enough data to really come to a conclusion here, so I will not cover this topic as well. I'll concentrate on the germline variants, the risk for CML, or on influencing therapy. The questions I'd like to address are: what do we know about familial CML or syndromal CML? So CML which occurs in more than one patient in one family, or syndromal CML which occurs in association with a clinical genetic syndrome. Second, genetic variants associated with susceptibility to CML. So are there genetic features with increased risk to have CML? And then genetic variants associated with therapy response. And we start with the latter one first. In general, and this is shown by this overview, there are many factors that possibly could influence the tyrosine kinase inhibitor therapy, and they include the microenvironment where the blood cells live, might include metabolic features, might involve immunological features, but I'd like to focus here on the genetically determined aspects, and in particular the ones which influence the pharmacodynamics and kinetics, so how the drug gets into the cell, how it works there, and how it's pumped out of the cells, because this does influence the effect of the TKI substantially. So we call this track transporters, where the track goes in and out the cell. And this is obviously an important point, because the track we just swallowed, the tablet, has to be taken up by the gut, is going into circulation, is metabolized in the liver, and eventually excreted in the kidney or the gut again. And a number of genes involved in these processes are mentioned here, and you will see them later on in the list of genes we examined. And the questions again we wanted to ask, or which have been asked in other studies, is how does these genetic polymorphisms, so genes or parts of genes which are different between people, whether they are associated with CML. And I must say, this is our data from adult CML, but has been found that certain polymorphisms might be associated with pharmacogenetics, so how the drug acts on early or late, or whether this influences the early or late treatment response depending on your genetic profile, or whether this impacts on imatinib levels, so how the drug is concentrated in the blood, whether side effects are dependent on these pharmacogenetic features, and then more general aspects like how the genetic features influence apoptosis, so the programmed cell death of CML cells, or repair features, or immunological regulation. And overall, there have been many studies, and we selected 32 previously described single nucleotide polymorphisms, or the combination of such polymorphisms called haplotypes, and tested whether they had an impact on, as I said, the treatment response or imatinib levels in children as well. This has been tested in different genetic models, and turned out that only in 5 cases, 5 of such SNPs, we had results consistent with these previously described single nucleotide polymorphisms. 4 had opposite effect, and 33 did not show the correlation described in the literature before. Now you would wonder, why is this the case? Is it the reason that because childhood CML is so different from adult CML? Or what is mainly or most often the cause? Are the numbers too small to really show an effect? And as I said, we tested this from the German CML cohort. These are about 300 children with CML, and this is for association studies a rather low number. But the mentioned studies here, the 32 other studies from adult CML, they reported about case numbers in the order of 50 to 230 adults. So this is probably also in this case rather a matter of low numbers. And we have different ethnicities which may have an impact on that. So overall, we cannot identify certain genetic polymorphisms with strong impact on the early or late treatment response or imatinib levels in children. One positive example where you can see such an association is this reporter gene, which transporter gene, which is involved in the transport of imatinib. It has been shown in this previous study that a certain type, haplotype, which involves this risk genotype T, is associated with imatinib resistance. So you would expect that they respond to imatinib treatment worse than the people with the normal gene or nucleotide. And this was reproduced in our cohort. So you see the patient with this risk allele, they achieve major molecular remission to a later time point than the others. And if you don't do this testing on genotypes only at one location, so at one nucleotide, single nucleotide, but on many throughout the whole gene, you can theoretically do what we call a whole genome association studies, where you test thousands of loci and see whether the disease is in some way associated more than just random with a certain position in the gene. And then you could identify which gene might be responsible for this susceptibility towards a disease, for example. And this has been done in many diseases. And again, usually you need large numbers of patients and controls to get this small effect visible in such a GWAS analysis. And we had only the 310 samples from the German CML registry and 4,000 control patients of matched children from Germany. They have been analyzed using this gene array. And again, as I said before, you usually need very large numbers of patients and controls. But in CML, there are only very few results from adult CML GWAS, and in this one reported by a group from Korea, they had again only 202 Korean CML patients, and then a validation set of another 200 patients from Canada, and they identified certain risk alleles or risk loci, and did follow this in more detail to pin it down to a certain genetic region and certain genes. So this is why we had some hope that we could identify something similar even in our small cohort of children, but it turned out that none of the loci reached significant more than random distribution above a certain level. And even if we combined this with these previous data from the Korean group, that did not reach significance. So we cannot identify a risk allele or risk haplotypes for pediatric CML overall. Interestingly, such a GWAS analysis from a French group, they had again relatively small CML group of 433 patients, and they did identify haplotypes or combination of polymorphisms, which was significantly associated with predisposition to CML. And interestingly, this risk score was associated with age at diagnosis, and the average number of risk alleles was significantly higher in younger patients. So which again somehow motivates us to keep going in this direction. And therefore, we believe there's reason to still try to discover germline variants with an influence on the risk of CML, but maybe on a different approach. And I'd like to show the idea we are following here. if you consider the epidemiological pattern of rare childhood tumors, there's a very general view that you could say there are tumors have a low incidence in general and occur exclusively in young age. For example, neuroblastoma. And there we know that there are germline pathogenetic variants identified with a high association to neuroblastoma. So the tendency, or the likelihood that you develop neuroblastoma is higher once you've got these germline variants. Then there are tumor entities with a more bimodal age distribution. And this is interpreted as an age-dependent biology. For example, acute myeloid leukemia, where I've got certain types which occur typically in very young age, even in infants. And then the incidence increases with increasing age again. And again, there are strong data of germline predisposition in certain types of acute leukemias and myeloid neoplasms. And this is why we argue, why should myeloid neoplasm in general and acute myeloid leukemia be associated with genetic polymorphisms, but not CML. Then there are typical entities of adulthood that rarely occur in childhood, for example, colon cancer, which is almost not present at young age, but there are some patients, and if we identify in these patients the possible genetic reasons, then it has been found that they carry to a high number cancer predisposing genes. So they have mutations in genes, for example, which work for DNA repair. And if this doesn't work properly, the risk of developing cancer is far higher. And the patients which develop, for example, colon cancer, they have to a high proportion such variants which tend to have the typically disease of older age at earlier age. And then eventually there's entities of adulthood with rare occurrence in childhood, but they have a very specific biology, and that's what we believe is the case for pediatric CML, but this has to be proven. And why do we believe this? Because this dynamics, these curves somehow mimic the distribution of CML over age. So the typical age in Germany at diagnosis is 65 years, and we have only a small number of patients below 18 years of age, but there's no particular bimodal distribution, but rather this shape, and this is somehow similar to these theoretical schemes. And this is why our hypothesis is that we are not expecting rare alleles, rare mutations, which cause an inherited disease, including CML, as you know from other diseases. But again, we were not able to identify common variants which have a small impact on the risk for CML, but are present in many, many patients. But since we have this BCR-ABL as oncogene, as a strong oncogene, we probably need only rare and common variants with work together and increase the probability to develop CML. And this point why I said that there's not much indication that strong inherited CML exists. This comes from reports on only 5 families with a total number of 14 affected individuals, which had the Philadelphia positive CML among several members of the family, including a pair of identical twin brothers, or sibling pairs, or a girl and a mother and grandmother. So some indication that the trait within this family would predispose to CML. However, this is relatively rare, obviously, and therefore it was concluded from this observation that it appeared more likely that instead of a simple Mendelian mechanism of polygenic mechanism led to a heightened susceptibility to CML in these families. And the same is true for clinical syndromes, so conditions where a set of features is combined with each other because of a certain genetic variant. And there are only very few examples that CML is included in such a genetic syndrome, and therefore it's concluded that CML is not known to be strongly associated with clinical syndromes. However, having said that, is that it might be the case that until now, because of that not that much genetic workup had been done for methodological and resource reasons, we could pick up more patients with such a condition if we would look closer to it. And for example, in Germany, we did identify a patient had this Noonan syndrome and this SOS1 mutation, which is in a mouse model associated to interact with BCR-ABL. So I assume that there are more cases than we know of until now. And as an example, we did analyze this in several families with twins and triplet, because we thought they are most closely related to each other, and it's easiest to find out whether this is a genetic variant associated with disease or not. And it seemed to be the case that certain alleles which might have an impact on the risk to develop CML somehow are collected within the individual, which eventually develops CML compared to the siblings which have not these alleles. This is not a proof of a genetic cause, but an indication, and prompts us to follow our hypothesis that there is no strong effect of genetic variations within CML, because we do have this strong oncogene BCR-ABL, but BCR-ABL only rarely hits a cell which eventually then develops towards CML. But if you do have a combination of genetic variants, as shown in these examples before, then the risk is increased that BCR-ABL hits the right cell to become CML in the end, and therefore increases the stochastic chances that BCR-ABL eventually leads to CML in these predisposed cells. And therefore, I'd be very happy if you, as viewers of this video, or your treating physicians, as researchers, which are aware of such cases of familial CLL or syndromal CML, would report or get in touch with us, because we really wanted to bring this forward and to identify how this could be explored and explain why some children develop CML at such young age and which impact this might have on the management of these cases. And therefore, yes, please do get in touch if you've got information on such cases. And with that, I'd thank for your attention, and I'm happy to see you in the near future. Thank you very much.